Hello everyone, I'm Mahfuzur Rahman from the University of Minnesota. Today I'm going to talk about standard-free benchmarking of replicated genome-wide CRISPR screens. Genome editing on a large scale is a powerful approach to learn about biological systems, and it has recently been revolutionized by CRISPR-Cas9 technology. To knock out a gene using CRISPR-Cas9, we need a guide RNA that targets a part of the gene. The Cas9 protein then introduces a double-stranded break in the targeted region, which is commonly repaired by error-prone non-homologous enjoining, introducing insertions to deletions and effectively resulting in a knockout. Now, to do this on a genome-wide scale, we make a library of guide RNAs and transduce the cells in a pool setting so that we can do parallel editing on many different genes, which is shown in the top part of the right figure. However, this will produce a single gene knockout for all the genes. To create double knockouts, we have to first introduce a stable gene knockout on the wild-type haploid cells and then perform a genome-wide second knockout using CRISPR. Now, to evaluate the quality of these CRISPR screens, one approach is to conduct many independent replicated screens and summarize the agreement of those screens to generate a gold standard. For example, to define an essentiality gold standard for single mutants, Margaret et al. Selected genes with significant essentiality scores in 50% of the 72 screens they conducted, resulting in almost 300 essential genes. However, the definition of gene essentiality is sometimes context-dependent. For example, there are tissue-specific essentials, and it may also depend on the specific experimental assay. Therefore, a data-driven definition of such a gold standard may be useful for evaluation of screen quality. Here, we will present such a method to tackle this problem. We divided the problem in three different steps. First, we need to generate genome-wide CRISPR screens for single and double mutants, and we need multiple replicas per screen so that we can use the replicas as independent measurement of the same signal, and taken together they can be exploited to reduce the error associated with individual experiments to generate a consensus output. And then we need to devise a computational method that takes advantage of these multiple replicate screens and formulate a consensus profile to use as a data-driven gold standard. And finally, we need to use this gold standard to evaluate the reproducibility of the screens. Here we introduce Jitter, joint estimation of data and error rates, that given a set of replicate genetic screens, computes a maximum likelihood estimate of the false positive rate or FPR, false negative rate or FNR, and along with the consensus data profile that accounts for these error rates and all replicate observations. Jitter takes as input a set of quantitative replicated screens and a set of priors for FPR and FNR. It then uses a Markov chain Monte Carlo approach or MCMC based approach to jointly estimate FPR, FNR, and derive a consensus posterior probability profile on the observed phenotypic effects. In addition to computing the consensus profile, Jitter also binarizes the consensus profile to use this as a gold standard. One way it does it is it takes the posterior probability distribution and applies a cutoff of 0.5, meaning any gene that has a posterior probability of greater than 0.5 are deemed essential or 1, and the rest are deemed non-essential or get a 0 in the gold standard. Then Jitter uses the gold standard to benchmark screen quality as a function of different data thresholds, for example, effect size or statistical significance or both. In our first application, we applied Jitter to estimate FNR FPR based on 5 independent single mutant replicate screens completed in human haploid cell lines. First, we measured the abundance of each gene in the library at a starting and ending time point, and the relative abundance are quantified in terms of log-fold change. And we have noticed that essential genes tend to rapidly drop out of the population and therefore exhibit very negative log-fold changes. We applied Jitter to derive maximum likelihood estimate of FPR and FNR for identification of essential genes or genes with a very strong fitness defect. Focusing on genes exhibiting at least a twofold change or log-fold change of less than -1, we applied Jitter to jointly estimate a consensus set of essential genes and FPR and FNR. Jitter analysis produced maximum likelihood estimates of FPR and FNR of 0.004 and 0.149 respectively, which correspond to a precision recall of 98% and 85% respectively at a log-fold change of less than -1. We further evaluated single mutant effects at a range of effect size thresholds and measured the corresponding precision recall characteristics. For example, at a log-fold change less than -2, we measured a precision recall of 100% and 26%. And also, we also measured a precision recall at a weaker effect size at a log-fold change of less than -0.5, where we get the precision recall of 61% and 98%. As expected, we get higher precision and lower recall at a very low or very high negative threshold. We also applied Jitter to evaluate the reproducibility of genetic interactions called from double mutant phenotypes, focusing on genetic interactions from Fas and query mutant. Now, genetic interactions arise when simultaneous perturbations in two genes cause a more or less severe effect than expected, given the single mutant effects. Now, when the combined effect is sicker than expected, this is termed a negative interaction. Otherwise, it is termed a positive interaction. Quantitative genetic interactions, we scored them for our screens by measuring both an effect size and a statistical significance FDR on Fas and replicates. As with single mutants, we evaluated the precision recall characteristics of genetic interactions called at a range of thresholds. For example, at a relatively stringent cutoff score, -0.75 GI score and FDR of 20%, Jitter reported a precision recall of 87% and 14%. We can see that the reproducibility of genetic interactions are much difficult than the single mutant effects. In summary, Jitter enables a standard for evaluation of skin quality for genome-wide CRISPR skins, and genetic interactions are substantially difficult to reproduce as compared to gene essentiality, and I expect Jitter will generalize to other genomic and proteomic settings to enable precise estimation of reproducibility. Thanks.